Energy production in cells. Overview of cellular energy production. Cells utilize proteins, fats, and carbohydrates to produce energy. Digestion occurs in two phases, extracellular, in the gut, and intracellular, within cells. Glucose is a primary energy source, converted into acetyl-CoA in mitochondria for ATP generation. Other nutrients, including fats and proteins, can also produce acetyl-CoA and ATP. The process of energy production is crucial for cellular functions and overall metabolism. Nutrient utilization. Carbohydrates are broken down into glucose, which is then converted into acetyl-CoA and ATP. Fats are metabolized into fatty acids, which can also be converted into acetyl-CoA. Proteins are degraded into amino acids, which can similarly be transformed into acetyl-CoA for energy. Organs like muscles and the heart can utilize both glucose and fatty acids for energy. The brain primarily relies on glucose but can adapt to use keto acids during starvation. Fat metabolism. Types of fatty acids. Fatty acids are categorized into long-chain, medium-chain, and short-chain types. Long-chain fatty acids require carnitine and CPT, carnitine palmitoyl transferase, for mitochondrial entry. Medium-chain fatty acids, such as those found in coconut oil, can enter mitochondria without carnitine, allowing for quicker energy metabolism. Short-chain fatty acids are produced by gut microbiota and have distinct metabolic pathways. Deficiencies in fatty acid metabolism can lead to serious health issues, including carnitine and CPT deficiencies. Health implications of fatty acid deficiencies. Carnitine deficiency can lead to muscle weakness, seizures, and fat accumulation due to inadequate ATP production. CPT deficiency affects the body's ability to utilize long-chain fatty acids, leading to energy deficits. Understanding these deficiencies is crucial for diagnosing metabolic disorders in infants and adults. Early intervention and dietary management can mitigate the effects of these deficiencies. Starvation, ketosis, and related disorders. Starvation and ketosis. During starvation, glucose stores are depleted, prompting the body to break down fats for energy. This process produces ketone bodies, leading to a state known as ketosis. Ketones serve as an alternative energy source for the brain and other organs, but can lead to ketoacidosis if produced excessively. High-fat diets can induce ketosis by overwhelming mitochondrial metabolism, resulting in excess ketone production. Carnitine deficiency syndromes. Carnitine deficiency can occur in preterm infants or individuals with genetic disorders. Symptoms include muscle weakness, seizures, and fat accumulation due to insufficient ATP production from fats. Early diagnosis and treatment are essential to prevent severe complications. Peroxisomal disorders. Disorders such as Zellweger syndrome, Refsum disease, and X-linked adrenoleukodystrophy, ALD, prevent the metabolism of specific fatty acids. These conditions led to the accumulation of toxic fatty acids, causing neurological and hepatic issues. ALD is notably depicted in the film Lorenzo's Oil, highlighting its impact on neurological health. Cellular degradation systems. Lysosomal and proteasomal functions. Lysosomes digest unwanted materials using enzymes. Deficiencies can lead to disorders like Gaucher's and Tay-Sachs diseases. Proteasomes degrade misfolded or damaged proteins, with implications in diseases like cystic fibrosis. Understanding these systems is vital for developing treatments for related disorders. Lysosomal storage disorders. Lysosomal storage disorders arise from enzyme deficiencies, leading to lipid or carbohydrate accumulation. Examples include Gaucher's disease and Tay-Sachs disease, which often result in neurological deficits and organ damage. Management strategies focus on enzyme replacement therapies, though they can be costly. Proteasome function in disease treatment. Proteasomes play a role in degrading defective proteins, including those involved in cancer. Drugs like Velcade inhibit proteasome function to treat conditions such as multiple myeloma. Research is ongoing to develop drugs that modulate proteasome activity for various diseases. Clinical applications and nutritional considerations. Ketone bodies and brain function. Ketones are a crucial energy source during fasting or low carbohydrate intake, especially for the brain. Excessive ketone production can lead to ketoacidosis, a serious condition requiring medical attention. Medium-chain fatty acids can rapidly produce ATP and ketones, beneficial for conditions like epilepsy but risky if overconsumed. Nutritional deficiencies in specific conditions like anorexia nervosa can lead to severe nutrient deficiencies, resulting in muscle wasting and potential organ failure. Management of these conditions requires careful monitoring of nutrient intake and metabolic status. Cachexia in cancer. Cachexia is a wasting syndrome prevalent in cancer patients, characterized by accelerated degradation of nutrients. Unlike normal starvation, cachexia involves significant protein loss, complicating treatment strategies. Treatments aimed at increasing appetite often result in fat gain rather than muscle recovery. Final notes on diagnosis and treatment. Understanding nutrient metabolism and degradation systems is crucial for treating disorders like carnitine deficiency and mitochondrial disorders. Specific testing and dietary adjustments are necessary for diagnosing fatty acid metabolism disorders in infants.